Welcome to another episode of the Dendroid podcast with Said and Nika. This is a conversation with Dr. Keith Wheaton. He's the director of Translational and Molecular Medicine program and an assistant professor in the Department of Biochemistry, Microbiology, and Immunology, Faculty of Medicine, University of Ottawa.
During his scientific career, Dr. Wheaton has investigated molecular mechanisms of aging and cancer biology, with a particular focus on cellular senescence. His work spans biochemistry, cell biology, and molecular genetics. His interests include cellular senescence and its role in aging and cancer, molecular biology, and biochemistry of disease processes.
We hope you enjoyed this episode, and now here's Keith Wheaton.
All right. Hello. Good morning, Dr. Wheaton. How are you?
I'm great, thank you.
Could you tell us a bit about your academic journey? How did you come about becoming a biochemistry prof and BMI prof?
Sure. So it's probably not going to be a linear story, but
Usually isn't.
Yeah.
maybe you've gotten that before. So I started off. I went to Guelph University from undergrad. I did a biochemistry degree there. I also did co-op there. I worked in a lot of different workplaces as part of that. Then one of those workplaces was a pharmaceutical company called Lelix Biopharmaceuticals. I was able to get a full-time job after I graduated. It was kind of fun. I got a letter in the mail that week I graduated saying that they offered me a job, so it worked out well. So I stayed there for about a year before I went to graduate school. And there I was doing basically migraine research, and they were doing drug development for migraines. And so I did a lot of cell work and things like that there. But I wanted to do a graduate degree, so I went to Calgary. So I did my PhD in Calgary. I spent about 8 years there, approximately. Also met my wife there, had a child there, and then came back. Essentially, then I went to do my postdoc in Toronto, downtown Toronto, with Dr. Benchmall. So he's famous for P53. And then I worked there for quite a while, actually. So, but while I was there, I transferred up to York University. And then I spent the rest of my time there for about, it's embarrassing to say how long I postdoc for, but it was about a decade. So, but we were very successful there. We got several grants, and so I'll probably be talking about some of that research a little bit later on in the interview as well. And then, I started teaching at York University. I did contract teaching. I extended my contract teaching out to McMaster University too, because I was living in Hamilton at the time. So I taught a lot of contract courses before I came to University of Ottawa. And then I was offered this. It was actually a year contract when I first started. And the other thing that they said that there would be on-site and off-site for essentially teaching. And so I had pre-interview, and the off-site ended up being in China. So I actually taught in China for about, 4 years. Well, like just for two months at a time, essentially. So I was there for a considerable amount of different cohorts that were going through. They basically kind of copied the medical curriculum that is in here in Ottawa, and they taught it in China, and there was a cohort there. And us and a group of other professors from the University of Ottawa were teaching pre-med, essentially there. So it was interesting, it was fun, yeah, so it's a long way to go for a course, but so that was kind of my origin. And then at the same time, we were developing something called TMM, which is Translational Molecular Medicine. So I'm one of the kind of founding professors in that as well. Given all my teaching experience before I came, that's why they kind of selected me. Oh, and there's I also did an education degree in that time as well, just before, it was kind of at the end of my postdoc, and then I finished it up just as I started working here as well. So I have a B.Ed as well to help me kind of facilitate everything that I've done in the program. So I think it's kind of what they hired me, and then it allows me to, do appropriate practices for the classroom and so forth. And I think I've done well since I've been here. So for the most part, so that's kind of the breadth of it. So the crib notes version of my educational and academic journey. So.
Gotcha.
So were you also you did mention that you worked in that particular company for about a year, and then after that you went to follow your graduate studies.
Yes.
Were you also keeping that in mind? I'm trying to understand, when did you make it? Was there a distinction that, okay, I'm going to work in academia, or I'm going to work in industry?
Yeah.
I don't think I had decided that. So one of the things I'm really interested in, at least in terms of subject matter, is senescence or aging. So cellular aging was kind of what drove me. And I think if students are watching this, I mean, you have to really go with what you're interested in, right? So, and that's what I was interested in. There wasn't that many labs at the time that were working specifically in that field. But there was one in Calgary. So that's why I went to Calgary. It's kind of random, but you may not, but you'll always be pleasantly surprised when you go to a fairly big city and look at what's going on there academically at bigger universities, because there's always really fantastic people that are at every place, right? So I decided to pursue that, because that's what I was interested in at the time. So, but that's not saying that the skills that I learned there at the pharmaceutical company aren't transferable, right? So that's where I started doing tissue culture and doing a lot of cloning and a lot of basically just general molecular biology, and that stayed with me. I mean, I'm really proficient at doing those type of things, and it allowed me to do a lot of the things that I was able to do in my PhD and my postdoc, both which were focused in cellular aging, essentially. So that's kind of why it came about. So, and it was just fortuitous that I got into a pharmaceutical company at that time, right? So that pharmaceutical company doesn't even exist anymore. So, but some of the drugs that they developed do. It's just they've been bought out by
the patent was
yeah, exactly. So I don't know about any of the drugs that we were developing. I think there's one that might be out there. I have to.
For migraine, I think.
Yeah.
Well, I mean, of course, I think they did quite a lot of other things, other than migraine. But for migraine, I think there are a couple of different packages. I think it's specifically marketed for migraine.
Yeah, absolutely there are. Yeah. So at the time it was sumatriptan, which was something big on the market then. I think there may be better derivatives now because that one had a lot of side effects. I look predominantly at serotonin receptors and then antagonizing those for various reasons of vasodilation and things like that. So sometimes I'm still interested in that, and I'll just look up a few little things because So there's actually some people here that work on it as well. So and Geo, so that have interest in migraine as well. But it's one of those things that you do become an expert in a few little things as you go along, but that may not be what you want to be an expert in for the rest of your life too, right? So
it's so interesting. The applications of working on serotonin receptors is so vast. You can hear like this, and then for depression and for quite a lot of other things.
Yeah. What's interesting is that they were actually working on some of the same kind of appetite suppressant things as well while I was there, because it's basically a neurobiology kind of section of the pharmaceutical company, and then I was just one group working on certain receptors, but some people were working on appetite suppression, and other people working on other parts as well for different types of drugs that they were interested in at the time for different conditions. So, yeah.
Very interesting. How did you discover your passion for senescence and aging?
I don't really know. I just always had interest in it. Actually, I think it goes right back to, I did a co-op in high school too, that I was really interested in, kind of premature aging syndromes as well. So I remember reading a book, and that became because I went to school in Hamilton, and I went to McMaster. I'd worked on a lab there as part of a high school part of it. And I was able to access their library, and I started reading up on certain diseases like that. And so that's when I became interested in it. And that, you might have an interest in that, but then finding somebody to work with may take a bit of time too, right? Because not everybody worked on that. I do. Actually, one of the bigs in the field was actually working at McMaster at that time. I wish I had known that because I was doing a lot of co-op during my undergrad. It would have been great to try to work in their lab at McMaster, but by the time I was looking for graduate studies, they had already skipped down to the States. So, which would have been, that would have been phenomenal as well. But I'm really happy with where I went in Calgary as well. I think it was a great place to go as well. So.
No, I'm asking this as we're going to have a class on biology of aging. But then what is aging biologically speaking?
Well, there's more than one thing. So that's the short of it. So it's not just one thing. So when I was young, I'll give you this little spiel when I'm in the course too. I felt like it was all genetically determined, right? So, and I've come around a little bit because there's multiple causes, right? So there is not just a biological determination. So to me, when I was looking at stuff when I was an undergrad, I thought, oh yeah, telomeres are everything, right? So, and while I was an undergrad, that came to fruition. Basically, that means that there was tons of data coming out at that time, and it was basically proven that that's the aging clock, at least for senescence. So maybe I should back up a little bit, and that a lot of this information comes from a few studies that were done actually a long time ago, in the 50s and the 60s, and it's something called the Hayflick limit for cells, at least for cellular aging, right? So, and basically what happens is that all of our cells have a limit of how many times they can proliferate. So, and then that's kind of defined by this researcher called Leonard Hayflick. And so he was able to kind of define that, reproducibly, cells can only divide so many times before they go into what's called a quiescent state. They don't die. They're still metabolically active, but they're completely refractory to any growth signals. And so they won't essentially kind of do anything for the most part when they're in that state. So the molecular biology behind that became telomeres, essentially, right? So, but at the same time, there was other research going on that kind of defined some of the other things. So the two competing theories are basically that program theory, that is due to telomere length and the erosion of telomeres. But there's also things like free radicals or wear and tear, right? So think of a car, it doesn't have cells or anything like that, but you're never going to be able to keep it pristine forever, right? So it's eventually going to break down, it's eventually going to rust, it's eventually going to decay, right? So that is what wear and tear is, right? And our bodies are machined too, and so they don't have unlimited capacity to regenerate either. And so that is part of the aging process as well. So there's two things. There's the, just the environment, wear and tear, and then there's genetic determinism. So it's easy to be a determinist when you're young, but as you get older, it's thinking, those other things, they have good data, especially as a scientist, they have good data, and then it makes a lot of sense. And then the genetic determinism part really only works in cells that can proliferate. They don't work in terminally differentiated cells, which includes your brain, which is a major part of a lot of aging diseases, right? So it also includes, atrophy of different kind of like your muscles and so forth. So all of those things don't necessarily have to do with, a genetic deterministic part. It has more to do with how your body starts breaking down over time. So unfortunately, we live in a universe that has entropy, and so that is what we have to deal with, right? So you guys are young, you probably don't have to think about it right now, but as I get a bit older, I get some gray hair here, a little bit of wrinkles, and now I'll have to really consider that, right? So that I can't avoid it, so you can try, right?
At least we can understand it.
Yeah, exactly.
And also there's this, of course, it's in the US, there's this one guy who's trying to stop the process of aging in a very crazy different
doing
ways.
Different surgeries and stuff like that to stay young.
Or a specific diet or specific exercises or treatments here and there.
So when you take the biology of aging course, I do a whole spot on diet and So, and there's lots of things there, right? So, and the students like it because it's like your way of being able to, maybe prolong your life. Or what's probably more interesting for the research part is trying to extend something called your health span. So at the end of everybody's life, you have a period of morbidity, which lasts approximately about 10 years. And you may know people like your grandparents or your, people that you've known along the way. Almost everybody experiences that. And it's where everything starts breaking down. Everything, and you'll find everything's going at once, right? They may have Alzheimer's, they may have heart disease, they may have, osteoporosis, and they're all contributing to each other, right? So, and that part of a morbidity is really what you want to try to target in some way. So I'm not trying to make people immortal or something like that. It's more you want to extend people's health span so that they can live, a healthier life up to closer to the end, right? So which is a much nicer way of thinking about it than, because if you extended life, but you just extended the morbidity part of it,
that doesn't really
it's not, the quality of life is poor, right? So, and then I can't even imagine the healthcare costs that would be associated with that, right? So you want to, make the quality of life much more, meaningful for anybody who's there, essentially, that part of their life. So
Makes sense.
Do you think just expanding the length of time, even with the morbidity, has been sort of the trend as well with all the hospitalization in the past, not few, but in the past like 50 or even like a century. So it's like the first thought it was just let's stay alive as much as possible. But then, it wasn't though, because like nobody knew that you're taking death out of the home, because not everyone, but the majority of people used to just pass away in their own home,
right,
with their families around it. But now there is this institution, there's this hospital, and you put everything there, and then there's these machines. So it's actually like they give people hope that this person is still going to be alive. But then after years and like cumulative experiences, then we see this side of it as well. That it's just more suffering.
It can be, yeah. So it really depends on the quality of life when you're in there. So I've recently experienced a lot of dealing with an old age home and so forth, or just long care facility, and it's hard, right? So because you do see I can understand from the healthcare professional side, because what they're doing is that they're just trying to make that type of transition, because it's just a matter of time,
yes,
right? When you get in there, like you're probably not going to be there for more than 10 years, most likely. So there's always exceptions, don't get me wrong, but statistically that's what it's at, right? So, and they know that, right? So, and then they can predict what's going to happen because they've seen it so many times, right? So, and it's hard as coming in as a son or a daughter, to realize that your parent is at that point, right? So, and it's just a matter of time, right? So you try to make the best of it too, right? So because that's all you can really do. It's going to be inevitable, it's even if it it may not happen when you want, but it's going to happen for when that's right for that person, right?
That's just life.
Yeah, that's just life, right? So and it wasn't as traumatic as I thought it would be, because maybe it's because I know that this is going to happen. I don't know, maybe it's predictable. It's not as much of a surprise. But then, when somebody passes away, you're not really, just thinking, well, that's the end of your story, right? So, and then, now it's time for your children to live the rest of it, right? So, yeah. So.
Okay. Going back to your research, you have mentioned P53 a lot. It's so special to you. Why? Where did this?
Once you go into a P53 lab, you'll understand.
Okay.
Yeah, it's a good question. Well, it does everything, so that's maybe why. So, because if you look it up, it's going to be hard to get a real definition on what it does and so forth. I tell people that they're coming into my lab that is like, don't look it up. Like, just let me kind of translate what's going on there, right? So, because there's thousands of papers, right? So, there's actually a whole conference on 53. right? So every other year, at least there used to be. I think there still is. Actually, I went to a conference that was just on mutant P53 as well.
Just
so
mutant type.
that was it. And so, yeah, they're very specific. So, but even with all of that, there's still a lot of actual unknowns, right? So, which is surprising to me, right? So you figure somebody would have like just dissected it completely molecularly, because it decides a lot of things, right? So it decides on whether you go into aging, decides whether the cell goes to apoptosis, decides whether it survives. As well. So it's like a cell fate decision. And that's just the tip of the iceberg. It's found that it does a lot of other stuff too, which is always kind of astounding to me that it can do so many things and still be properly regulated when it needs to be, right? So it's more of I guess it's more of a puzzle, and maybe that's what drives people to kind of look at it as well. It also can be an easy crutch if you're doing research and you find, oh look, there's P53s involved. Yeah, and that's not surprising, but it's easy to put it all on it, right? So as well. So in the lab, when I worked in a P53 lab, there was a lot of different types of projects that people were doing. A lot of them had to do with apoptosis, a lot of them had to do with survival or metabolism. You get a kind of a better breadth of all the things it can do. And so you want to see how it works in your system and how it's particularly, engaged in your system and how it might differ from some of those other things. So that's kind of what my research is kind of geared towards, to understand, because so maybe I should explain what it is, basically. So it is actually what's called a transcription factor. It binds to a lot of different promoters. And the promoters are on genes, so they regulate gene expression. So you can think about it when it invokes a certain response, like senescence or which is pretty much the same as aging, or apoptosis. It has a different kind of selection of genes that it invokes. And so what happens with those is that it will tell the cell to do this. Like it'll, when it's time for apoptosis, it'll upregulate a ton of apoptosis genes. When it's going to initiate senescence, it upregulates a ton of genes that are involved in cell rest and all the things that kind of lead to senescence, essentially. But it also can be involved in a lot of other things. So, for example, it can also be, and why it's called a tumor suppressor often, a lot of different things that are part of the tumorigenic process, it also suppresses. One of those is angiogenesis. And so it upregulates a bunch of genes that are involved in angiogenesis. So that are a part of that regulation as well. And recently, but I always I try to just stick with all those, especially in the courses, because there's other things too, right? Recently, I was doing a little bit of reading as well, and it's involved in something called pharamatosis, which is part of how your brain actually eliminates cells as well. So, and I think it would be too thin to actually do research on that, but if I ever, I do have some interest in looking at that as well. But those are, just some of the examples where it's actually, involved, and it'd be interesting to be able to molecularly dissect how it chooses those things. Mm-hmm. I think that's really where my research and why I'm doing it is because you realize when you have certain results, then P53 is responsible, and then you realize nobody really knows how it selects those things or why it does those things.
That was actually my following question. Yeah. How does one transcription factor become so diverse?
It's actually also a family, so that makes it even more complicated, but I usually don't mention that until later on when you guys are teaching also a course in genomic instability. Mm-hmm. There's 2 different versions of it as well, usually involved in it can also be involved in the same types of things, but it's usually involved in something like differentiation or survival as well. So these other versions of 53 as well, because we have we don't have 3 copies, but we have 3 different homologs of 53 in our body as well. So actually, interestingly, and if you think about bigger animals actually don't get cancer, right? Even though they have they do, but at a very low rate, like an elephant, for example. So an elephant has actually 20 copies of 53.
Oh, that's why.
Yeah. So there's a direct correlation between tumor suppression and how active p53 is in eliminating cancerous cells or causing them to go into senescence.
Could p53 be targeted as a potential therapeutic strategy to target cancer, possibly?
Absolutely. So, and often, there's a couple ways of doing that. So, one of them is to try to upregulate p53, right? So, in order to do that. So, there's a drug that interrupts its basically it's degradation, right? So normally, since it's so potent, it has to actually be really well controlled, right? So all the p53 being made in your body right now is actually it's already a protein, but it's being degraded. And it's being degraded by a ubiquitin system, basically, right? So there's a drug that interrupts that system and stabilizes p53 to allow it to do its job. Now, unfortunately, a lot of cancers, they are mutant in p53, and so it's not acting the same way. And so those are a bit harder to do. But it's not impossible. Those are probably more conventional. But on the ones where, say, it's just being degraded all the time, for example, that's one way of doing it, is trying to upregulate that as part of a strategy, more or less. Interestingly, you guys can take it too, by the way. I developed a lab for p53 as well in TMM. So, and anybody who's watching, you can take it as well. I'm going to open it up to everybody, not just TMM students, where you take HeLa cells, and they actually have a suppressed p53 as part of the reason why they're cancerous. And the reason why is that they are chronically infected with HPV. So HPV produces a couple proteins that actually suppress p53, right? And so that's one of the reasons why they can't detect damage. That's why they can't go through a cell fate decision. And so that's why partly they're cancerous. So what we do in the lab is you take HeLa cells and we use something called an shRNA to knock down those proteins, those viral proteins that are constitutively expressed in those cells. And lo and behold, 53 comes up. It's really cool to see because it's wild type p53, right? And then it causes apoptosis in those cells because those cells are really damaged. Like if you take a look at a HeLa cell online, look at its karyotype, you'll realize that it's not a normal cell anymore. It's got a lot of mutations in it, a lot of duplications, deletions of whole chromosomes. And so when you see p53 come up, and it causes basically apoptosis, right? So that's another way of doing it. So you can try to reactivate p53 somehow. That kind of research is kind of almost moot now. And the reason being is that there's a vaccine for HPV now, so it's easy to prevent. And it's actually one of the really nice stories about cancer treatment too, right? Because you can actually prevent some cancers with a vaccine like that. So it's one of the only viral cancers that humans get. There's a few others, but very few in total. But yeah, so if you're interested, that's what we do in the lab. So so you basically take you transfect the HeLa cells with that plasmid. You suppress those proteins, and then p53 comes up. What we do is we synergize that with another chemotherapy agent as well, and then you can see whether or not the cells go through apoptosis. So we do that with both Westerns, and we do that with immunofluorescence as well, because it's a good way of seeing it.
Is this one of the advanced method classes, or no? It's like a whole.
No, it's actually a whole class. So, and a colleague teaches the other half. So half is in the lab, and half is actually doing data analysis and like looking at papers and how to design experiments and things like that. So,
What's the name of the course?
inquiry-based research. So it's 3301. So that's a third-year course, but yeah. So there is some plans to expand TMM. And if that happens, this will be one of the core courses that we'll actually have. So that's why I was developing it as well. So it's evolved over the years. So it used to be like a gap filling course during pandemic, so if people wanted to come to the lab, but it wasn't necessary, right? So it's something to get a little bit of lab experience, and so it's evolved since then. So it's become, if we expand, it's going to be required with, what we'll probably do is we'll do C-phages in the fall, and then do this course in the winter. So,
cool.
Yeah.
Before we get into teaching, because there's a lot to talk about in teaching, but like just in this particular thing that you mentioned that the disruption of the interaction of p53 and ubiquitin.
Yeah.
Are we disrupting just the interaction of the two, or no? We're just disrupting the ubiquitin system in general.
You're interrupting the actual interaction between p53, so it's a very specific drug.
So well, if we disrupt the whole system, that does quite a lot of other stuff
exactly.
as well.
Yeah, no, you're just, so the way, so it's a enzyme called MDM, well, it's called MDM2. Basically, ubiquitin systems have three kind of components.
Is it the E1, E2, or E3 that we are?
Yeah, so it's the E3.
E3. Okay.
Yeah, so the E3 is what kind of what targets specific proteins.
Yes.
So essentially, what it does is it will hit P50. Well, it will bind to P53 on a certain region, right? And then it ubiquinates it on the C terminus. So it's like it binds it on the N terminus, and then ubiquinates it on the C terminus. It's a polyubiquination that is then recognized by the proteasome. Yeah. So, and then it degrades. So you're basically, it's a drug that specifically interrupts that interaction, essentially. So it's been designed so that it will basically kick MDM2 off. Now, when P53 gets active, it naturally does that too, right? So it, when there's a stress that comes in, it'll lead to different signal cascades that will essentially phosphorylate P53. There's some other methods too, and then there's a few other post-translational modifications in there. I just look at phosphorylation. But in that region where MDM2 binds, it ends up phosphorylating in there, and that's what kicks off MDM2. So that's how it's activated normally in our cells. So this is like an artificial way of doing it. So just imagine in a cancer cell that's maybe some of those damage repair pathways or signaling pathways are aberrant, right? So it's not able to signal to P53. So, and that's one possibility. So as long as you don't have mutant P53, then it should still work. And so that's why those drugs kind of work. There's also another strategy, because P53 is involved in a lot of things, right? Sometimes it can gain functions.
Oh, like with the mutations?
Yes. Okay. And then, so the strategy there is to actually inhibit it, which is maybe counterintuitive.
Yeah.
But it really depends on the cancer, right? So like if you get, so one of the gain of functions is something called mesendermal to epithelial transition. And so when that happens, it makes your cells more motile. And that's part of the process which makes cells aggressive in cancer. So P53 actually can suppress that to some degree as well, but when it becomes a mutant P53, it can contribute to that process as well. So it depends on the cancer again, but yeah, so that I don't usually mention that one because it's counterintuitive. We used to work my lab that I worked with looked at that too, right? So they try to suppress p53 as part of a treatment strategy for certain cancers. Yeah, so
it's very interesting, and it's
Yeah.
like I keep having now questions about this. Like, how does it also have? I mean, of course it does, but how is its interplay with the immune system, or does it even like to that degree also?
definitely. Yeah. So probably on several ways. I don't know. I probably don't even know most of them, to be perfectly honest. What I do in some of my courses, we actually look at how it interacts in response. It's like part of an innate immunity for viruses, actually.
Mm-hmm.
So that's one of the reasons why HPV inhibits it.
Oh,
Right.
Ooh.
Because
it's a mechanism to survive.
exactly. So, and you can name a bunch of viruses that actually have components that suppress p53. So that's how it's probably involved, right? So basically, because cells do not like double-stranded RNA, for example, right? So you'll activate, there's a p53 partial response that will lead to, the destruction of those things, right? So, but they're similar. They produce viral proteins that will actually suppress those things too. So that's one example of it. There's probably others too that I'm just not aware of. I'm not big on immunology, but yeah. So I'm sure it's involved very similarly. So some of the other ones, I think, there's a bunch of different viruses. If you take genomic instability with me, that's one of our little things that we do. You guys can research one, basically, and figure that out. Yeah.
What are the different types of research done on like a protein like this, like a transcription factor like p50, that's involved in quite a lot of things? So I can imagine like one could just study its like isolated interactions and because it does a lot of things. So one could study all those little things. One could study all like the big picture of the interaction, the regulation, like the modeling. I think must be like some sort of modeling also be involved somewhere.
Yeah, it would be nice if that was the case. So actually, I've always been thinking that the Shangri-La of that field would be to actually crystallize 53 because it has never been done.
Oh wow.
Yeah, so I've had thoughts about that, so I've talked to GF too. But there's a lot of problems with it because it's something called an undefined structure that can't crystallize essentially. But I think that's why it's so malleable as well, right? So because I think a lot of the post-translational modifications kind of lock that in, right? So and that's how it selects, right? Yeah. So what was the first part of your question again?
The first part is like how are the different ways that we can approach understanding its function or its interaction?
Yeah. So that's the other place I was going to go to with a lot of difficulty. So yeah, when you set out, you're usually the way you do it is like, oh, okay, I'll do the stress. We'll see what p53 responds to, and then we'll look at the outcome, right? It seems pretty straightforward on paper, but there's a lot of other things going on, right? So, like, what if that cell constantly has DNA damage? Replication causes DNA damage. All kinds of things cause.
How can you isolate it to that?
That's exactly it. So part of the reason that's why I like to work in normal cells, right? So because you can reduce the stress. Right, so, but sometimes you got to do other things as well, right? So, one of the systems that I worked on is in progeria, which is a premature aging syndrome. Those cells are, they're basically from different patient skin punches and stuff. They're what are called fibroblasts. They're mesenchymal cells that are easy to grow in culture, but the problem is that tissue culture itself is a stress. So when you put your cells in there, and we have 20% oxygen in our atmosphere, right, or 21 approximately, and then our cells never see that much oxygen. So it's like you're there in a hyperoxic environment all the time. So it was problematic, right? So So one of the things I did when I was doing progeria research is I actually got specialized gases that we put it down to what's called a normoxic condition, so that I could eliminate one of those variables. I don't know if I eliminated every one of them, right? So, and the problem is something. It's almost like I'm not gonna say it's like quantum or anything like that, but sometimes when the methods you use can also
Determining
what's going to. So the only way to really get into normal cells reliably is to use a virus, and as I just told you, P53 responds to viruses. So hopefully when you grow it long enough after, it won't have as much of a P53 effect, right? So, but that's the only way I can like suppress certain genes and upregulate others, right? So reliably in a normal cell. So it's a challenge, right? So you have to have a reasonable expectation that these are this is the only stress I'm looking at, right? So I have a cell line that I tested early on in my postdoc that seems to just have telomeres as their main response. So I use that kind of reliably to go forward, right? So to test different hypotheses. But, I got to be careful of what else goes on in there because it's still in a 20% environment. Of oxygen. It could, experience stress in other ways as well because of the culture media. So you have to be careful what you do.
Understanding all the different variables. It's
you won't. That's the thing, right? So you do your best, and hopefully that'll work, right? So. Because actually, one of the things we used to work on in the lab too, and I didn't work on it specifically. Well, I helped on the project. It was there's metabolic stress that P53 actually responds to as well. So I don't know if you've done tissue culture before, but if you forget to feed your cells or something like that, or like your glucose goes down, P53 might respond to that. So that's something you want to avoid, right? So so it can happen, right? So you want to make sure that that's not the case, right? So, and the other thing that tissue culture is kind of like I'm kind of harping on it, but it's not a normal condition under which cells normally grow too, right? So another thing that P53 responds to is oncogenic signals. So an oncogenic signal is something that is normally growth promoting, right, but is constantly on, right? So probably the most typical one you can think of is something called RAS, like it responds to growth factors, right? So if you introduce an oncogenic RAS into a cell line, P53 gets activated, right? So it'll try to shut the cell down, right? So because that's a response, it's another way that it responds to things, right? So just think you're in tissue culture, you're constantly throwing growth factors on these cells. It's like it's an oncogenic response almost. So you have to be careful of how much serum you put on there and things like that. So that you're not, invoking another response rate. So, so yeah, you have you can do your best, So, and sometimes I find that, especially with my last, thesis students that I had, I said, this looks really good on paper. It should work, right? And then it's like, that didn't work. and it's because, you can try to plan for everything for P53, but you can't. You don't. Yeah. It'll always surprise you, basically. So.
Makes sense.
Yeah. So.
All right. So switching gears a bit.
Sure.
So you have mentioned, we can guess that you prefer teaching, but between teaching and research, which one do you prefer?
Well, I think they inform each other, so I think it's important that I do a little bit of research so that it informs my teaching. And I stay kind of current as well, so I'm not really doing you any favors if I, don't teach you kind of the way to interpret science now, right? So, and rather, I did my thesis a long time ago, so, well, I did do postdoctoral work too, but that's still, I'm out of the loop, so to speak, if I didn't go back in sometimes and try to do a few things. And that's more recent too, because my job right now is predominantly teaching, right? So, and I just do research to just kind of stay a little bit current. And then, there's things that didn't work, or they just took a long time to do, because aging research sometimes takes a long time, that I'd like to see complete too, right? So, because they were funded projects, and I'd like to see those, eventually see the light of day. And there's some interesting results in there that I think the scientific community should probably know about as well. So I don't know if I can choose between those, right? So, because I do like being in the lab and like doing the research as well. But, I've obviously switched my career since being a postdoc to actually doing full-time teaching. And I know I enjoy that too, right? So, but I like to do it in terms of more of a mentorship idea. And TMM is the perfect venue for that, right? So that way I can, help you along on your career path to go somewhere where you want to go, right? So, and if you learn a bit about P53 on the way, awesome.
That's just added bonus.
Yes.
So how are students different from here? Because you mentioned like you in that project, the whole curriculum here was replicated in China.
Right.
What is the difference in how was the experience in general? I'm very curious.
Sure. So they were super smart, like definitely on the upper end. So I don't know if what the exact selection process is, but you can imagine there's a lot of people in China, right? So, and they're, actually, right off the bat, I should mention, their first choice isn't medicine like you guys. It's, going into finance or going into like international relations or something like that. Yeah. Because there's a lot of, I don't know if they're urban legends or not or something like that, but I'm not sure if the conditions working in medicine are all that great there. You think about us and how we're dealing with public health, right? It's much, much different there, right? So you have a lot of people going into medicine. And what happens is that, if you're seeing patients or something like that, it's maybe 5 minutes, 10 at tops when you meet with a patient. And that's all you do, like all day, right? So, and then one of the urban legends is that if that person dies later on, then their relatives are going to come and do awful things to you or something like that. So it's kind of a different kind of feel about it. So I didn't get that too much in the classroom necessarily, but I think that's one of the concerns, right? So, and I saw all kinds of different people come in, just like I would here for the most part. I mean, they knew they were going to be in a medical program, but Chinese medicine is set up a tiny bit differently too, right? So generally, you get into medicine out of high school. So I was dealing with high school students essentially, even though we were doing pre-med. And so it was a little bit different in that way. So I felt I had to like slow down as well because I talked too fast in the classroom. So I slowed down to about half pace, and it was really.
I think there was also like a sort of a language barrier as well.
They all spoke English pretty well. Actually, I think that's maybe one of the selection criteria. So the written part of it may not have been as good, but I think often they were translating. So that's kind of why I did it that way, right? So that was probably the biggest difference. In terms of academics, I think they were just as good as the highest students here, right? So at least the people that were in my class. There were some exceptions, right? So, and they tended to come from not mainland China because there was a few students from Macau, there was a few students from Hong Kong, and so their education system, at least in Hong Kong at the time, was a little bit different, I thought. And so they were a little tiny. Some of them were a little bit weaker in that respect. So because they probably weren't used to that type of system. I kind of shook it up too. So I did teach like a traditional Chinese way. So I saw a couple classrooms and it's like, wow, yeah. And so it was just basically the teacher lecturing at the front. Like you can imagine, like most of your probably first year undergrad was like, but the students just weren't paying attention. So they just, they didn't care, right? So, or they just thought, I'm going to study the notes anyway, right? So, or they're doing, doing whatever they're doing on social media. So it was a bit different, but I kind of like did some active learning like I would do in your classrooms too, right? So like I did discussions and I did, so you took metabolism with me. I did exactly that in China. So, because I taught metabolism there as well. I also taught basic biochemistry as well, but I did it in a very similar way there too. So, so it was different, but the students were very delightful and they're very, I tried to engage them as much as I could. It's easier in person now. So I did, the last time I taught there was basically online during the pandemic, and it was not great. So I really need to, that's the classroom that I need to be there, right? So, I learned a lot during the pandemic too, so I ended up recording those lectures and just sending them over. I don't know if they were happy with that or not, but you know what? It's funny, they recorded me every time. Mm-hmm. So I don't know, I might still be teaching in China. So they have lots to choose from. They got like about 5 years of video recordings, so I'm sure they could probably hobble something together. So.
You also taught a lot in Canada in different institutes. You mentioned York, McMaster, Ottawa. How do you find different institutes being different?
I did different. Probably the main difference is in York. There was a really wide. Well, I taught bigger classes there too, right? So I taught biochemistry. I hate to say what I called that course, but it was tough for students, right? So because there was a lot. There was like maybe. I taught it on the off season, which is about 120, but then I taught it once during the main sequence as well, which is about 400. And it was just like, it was difficult because it's like you need that mark for med, right? So a lot of students struggled as well. And I'm not sure if I should say the policy for York, but it was, I couldn't have more than 30% A's in the course. So, which normally may not be a problem, but with that so much invested in that course, a lot of people had problems with that as well. So, in order to design it to meet those requirements, it was tough, right? So that's why it was just a very different kind of atmosphere. A little bit adversarial, a tiny bit for some of those students, but but I taught very similarly to what how I teach now, right? So it was just different, right? So it's so nice to come here and have such small cohorts, right? So, I mean, it's night and day, basically, right? So, I recently went back to a big class like I taught first year physiology here last year. I taught 2 sections of it, actually. So it was actually okay. I remember it being a little bit onerous, but I really liked it. The students were really good. They're really engaged.
Like anatomy and physiology?
Yeah, so
on the main campus?
AMP 1105.
Oh, okay.
But that was our anatomy.
Yeah, you missed me by a year.
Yeah.
So I taught the virtual one, which was 400 students, and I taught the one that was in person at CRX at 300 students. So that was my recent go back to like first year. So I'll be teaching the virtual one again this year. So it was fun. And I don't know, I have a different perspective of it now a little bit too, right? So it's good to be in one of those fundamental courses that students have a lot of trouble with. So, and I feel like I have a lot to add to there as well. So I don't know if I can keep on doing it for physiology, but it'd be nice to be able to do that, right? So to remember those roots, right? So because with TMM, I mean, the students are very motivated, and we have very small class sizes, so it's a very different kind of atmosphere as well. It's great, don't get me wrong, but when you guys are usually on board with everything that I might be doing, right? So it's very nice. McMaster was kind of weird. So it's not the students or anything like that, but what I taught there. So it was a weird part of my life where I wasn't really employed anywhere else. So so what happened is that I was desperate for any course they would give me. So I taught a bunch of things there that I didn't expect I would ever teach. So one of them was health in the environment.
Okay.
And it was a geography course. But you know what? I had such a good time taking that course. It was so much fun to be outside your kind of, I don't know, I guess it was outside my comfort zone a little bit. But at Guelph, I did a little bit of environmental chemistry, and I did and so I really leveraged that. So everything I covered in that environmental chemistry course, I taught in that course as well. The other thing that was part of it, and probably the reason why I got the job, is because it was partly epidemiology with cancer clusters. And so they saw me as somebody that might know about that. So I did learn a lot to teach it. So that was fun too. And you know, I loved teaching that course because it was like a different subject every time, right? So I taught like hydrocarbons and the atmosphere, and I also taught like, you know, how viruses spread and things like that. So it was just very different, right? So and it was you know, it was a course that was shared by a lot of different students too, like in environmental chemistry in themselves, or like in you know, people in natural science and also in geography, right? So as long as I threw a map in there, it usually worked out. The other thing was I taught nutrition there, which I didn't expect I would do. So that's, you know, but I realized that it's basically just biochemistry, right? So and that's why you've experienced how I teach nutrition, or I should say metabolism. I put a lot of nutrition in there too, and that directly comes from my experience in doing that course as well. Because it was fun to teach, right? Because you get...
It is a nice touch to it. We loved it.
Yeah. So I think that really adds to it. So that was a big course too. That was like, excess of 400 students, right? So, but the biggest course I taught there was actually genetics. So, and I don't know about you, but you know, every course is a bit of a challenge, and sometimes I have to relearn a lot of stuff. And they didn't give me it was just half the course. They didn't give me the second half, which is all the molecular biology stuff. They gave me the first half with all the Punnett squares and stuff like that. Oh my gosh, that was the top. But I learned it all. I did, and I thought I taught it okay. So, so I was doing stats and stuff like that, and polynomials and things like that. So that I probably wouldn't be able to do it now. But what was great about that course was that there was an instructor there that I was replacing for just the semester, and she did everything in terms of cat genetics and cat coding.
Oh, we have, like, do you remember? I forgot, Dr. Montpetit. He had different cats.
Oh yeah, sort of like just as a manifestation of different phenotypes and how can we like talk
exactly.
about that. Yeah.
I thought it was a great way of doing stuff.
Yeah.
So I learned a lot about cat colors and calicos. And
Yes.
so I thought that was fun. So that's what kind of made it fun anyway. So I tried to do a good job at that. So and I actually was teaching there, and I ended teaching there for the first half of that semester, and then I came here. So that's the last time I taught. So yeah. So those are kind of the general stories I guess I have for those courses.
There's definitely a big difference between small classes and big classes because like after like I don't know, after 60, 70 people, it's just you don't know anyone in that class, and it's just you versus just the material, and there is no human interaction.
Yeah.
It was tough the first year. The second year, it was like, well, the least I can just sit in front.
Yeah.
So then that sort of replicates the first like couple of rows. Then it becomes it's somehow like a small court, and then people who don't really want to interact, they just go sit
At
like
the
up.
back, yeah.
It's true. Yeah. So and I tried some, because I was doing my education degree at the same time. I tried a lot. I taught I cancer course at York as well. It had a limit of enrollment around 100, so it's not too badly sized, but I did a lot of active learning in there. So, and I think it took the students a little bit by surprise, but some of them loved it too, right? Some of them were right into it, some of them didn't want anything to do with it, right? So, probably the biggest thing I did, and I still do it actually, is I make them do a public awareness video, and which I think really helped them kind of, understand the material a little bit better and then have fun doing it too, right? So, I mean, cancer's not fun, obviously, but they're, you can engage in a little bit of creativity too, and then it's not, me just going over pathways or something like that, right? So, because that's about the last thing you want to do in a course like that. You want to generate interest and generate true compassion for like the disease and its many forms as well. So I teach that course here too. I just teach the first half, but I incorporate that video, for example, in here as well. So, yeah.
Taking your course in metabolism, I think you aspire students to understand the pathway, how it's regulated, rather than just memorizing the names of the enzymes and stuff like that. How do you encourage that?
Just by saying it. Well, I think it's probably an easy sell for the most part, because students don't want to memorize big, long things. And then to me, it's like, so why are you taking this course, right? So for me, if it's, I know most of you are taking it because you want to go to med school, but for the most part, I want you to come away knowing a little bit, right? Or that you can actually apply some of your knowledge after you come out. And that's the whole point of doing a degree to me, right? So to me, I'm doing a disservice if I'm just making you memorize a bunch of stuff that you're going to forget after and nobody really cared, right? So at least there's a chance that you can remember, oh, I remember, Dr. Wheaton talked about this thing in class, right? So, whether it's like the wallpaper or it's something like that, those analogies or those kind of little stories kind of solidify what that was about, right? So that doesn't always work. It's not going to always resonate with all students. Sometimes it doesn't work certain years too, right? So sometimes, but that's the nature of education as well, right? So you try a bunch of different things. Sometimes, I've done other things in the past too, and they didn't work, and so I dropped them or changed them or modified them. So it's an active process for me too, right? So because I want to try to maximize engagement as much as I can. For example, in that course before, I used to have the food science a little bit in there too, right? So I went through a little bit about how brie and stuff like that, and the fungus on the top, and all the biochemistry involved in that. And it's just like, I'm not sure if people were that interested. So it depends. Maybe if somebody's into molecular gastronomy or something like that, that would have been great for them. But I think for the most part that didn't work, right? So, but I thought it might, right? So I gave it a try, and I did it a little. There's maybe still some remnants in there, like my caffeine or something like that I have in there. But that serves more a bigger purpose, right? So that, that's kind of the way I do it, right? So is through those kind of little stories or those little kind of tidbits of information that you may not know about that we can expand and you can explain with the course. So if I was able to do the biochemistry course too, there's actually a much bigger one in there that spans over several lectures, because I talk about cholera at one point, right? So, but to understand cholera, truly understand it, you need to have all this background information, and I cover it in little sections all the way through, and then I tie it all together, like the receptors, how diffusion works, how and then you understand why it works, and then you understand what the cure is and how easy it is, right? In terms of the biochemistry as well. So I love doing that as well. So that gives you kind of this unifying way of understanding it, right? So that's kind of the way I like to do it, right? So, and then, if you're a doctor in a place where there's a cholera outbreak, you're going to suddenly go, wait, you talked about this.
Yes.
It's just sugar and like salt and fresh water. I can do this, right? So it's an easy cure, right? So.
Particularly because for a course like biochem, it's like just studying the nuts and bolts of everything that you're going to need later. It's like studying algebra because you're going to have calculus eventually. Like on its own, it's not, okay, well, these are the different types of sugars, how the structure is made, okay, what's the other building blocks, like nucleic acid and the proteins and amino acid. But at the end, it's just, okay, so it's just this huge like amount of information that will eventually come together because you're going to need to understand those if you want to study anything about.
But if you don't purposely, as an educator, bring those together, the students won't.
Yeah.
So for the most, at least when you're at the second year level, even third year level for the most part, you start doing it in fourth year a little bit. But and so that's. TMM is kind of more designed that way, right? So I don't apologize about it. I just, we're going to do the mechanisms and how it's regulated, right? So, and you might have noticed that there's a progression, I think, in some of those courses too, right? So that, and that's largely intentional. I mean, sometimes it has to do with instructor too, but I start with real basics, right? So, but then, for example, the metabolism course, the other instructors, they have much more specific information, right? So, but I feel that you're ready for it too, right? So, and the same thing happened in molecular biology, as far as I understand as well. So that you start with kind of the basics and the nuts and bolts, and then you kind of tie those together in later parts of the course, right? So. And I try to do that with, other courses as well. So the biology of aging is a good example as well. So we have two halves of that. I mean, we do molecular biology, cell biology in the first half, essentially, and maybe some genetics as well. And then in the second half, we do all diseases, right? But in order to understand those diseases,
you have to understand
a lot of cell biology, molecular biology, and we do a bit of physiology in that second half too, right? So because that's part of the aging process as well. So all of these things, so it's a way of what's the scaffolding the information that allows you to, kind of learn it, not necessarily in a. Everybody learns differently. That's why I'm kind of trying to choose my words kind of carefully and stuff like that. But it just creates basically a template in which you can at least engage with a little bit. Everybody's going to do it a little bit differently too, and I realize that. And I think appealing to the way people learn in the most diverse way is the best way of doing teaching and learning. So, yeah.
You have taught a lot of different subjects. Which one is your favorite one?
Oh, it's biology of aging for sure.
Yeah. Okay. Then we're gonna see how you teach that course in the fall.
Yeah. It's upcoming.
Yes.
Yeah, it truly is. So yeah, I think that would be the best one. It's funny, my history with biochemistry and stuff like that. So the reason why I can, I teach metabolism now was just because at York, that was one of the first courses that came available, and I was able to teach it, right? So, but I had to learn everything again. I don't know about you, but I forgot all about biochemistry by that point, right? So I learned it all, right? So the entire textbook as I was going through the first time I learned that course. So now you get the the revised version, I've taught it so many times now. It's probably about 20 times now, actually. So yeah, so you get the kind of just the metabolism part, but in China I taught a lot of the biochemistry as well, and then I taught biochemistry and molecular biology at York. See, it was a tough course. I actually had three subjects in there. I had basic biochemistry, like all the macromolecules. I did metabolism. Not everything that we did. And I did molecular biology.
There's not enough time.
Yeah, but it was. That's why it was a tough course. So that's why the average was where it should be to get 30% A's. So it's partly designed, right? So, but I hate to do that because it's not fun to do, right? So that's why it's a little bit different, right? So if you have to design it to get a certain average or something like that. So it's so much more fun in TMM because, man, whatever happens, happens, right? So if you got an A, you got an A, great. So.
All right.
Well, I think as the last part, we can just talk about TMM in general. So you mentioned that you were one of the founding professors.
Yes.
We have asked about how like TMM came to be and just like what is it? What's the difference between TMM and many other programs that are considered considered pre-med, but I don't think that's the best way to like describe them. But a way to understand how human body or how just living beings works. So, what's special about TMM? What gathered all this group
Also,
of
we
people
also
together?
have CMM here. So
Yeah.
how is it differentiated?
So TMM is strictly an undergraduate program. So even though we consider ourselves part of, the TMM program, obviously because we're instructors in it, right? So it's actually the founding members are 3 different departments besides TMM, right? So that's CMM, that's BMI, and that's the EPI department as well. So although they don't teach as many courses, but they've been changing that recently. So there's some advanced methods I think you can take as well if you're interested. So they put together a proposal for this, kind of led by Dr. Cochier. I don't know if you know who he is. So he was the first director of TMM. And so they were trying to put together a new program that was research-oriented, used modern teaching methods, had high faculty ratio, just so they could do something different than other programs would do. Because when you start an undergraduate program, you really need to stand out as being a bit different as well. But then also, it needed to serve people who were interested in career development as well. So that's kind of why we have everything that you require for med school as well. But I think you probably get all that in first and second year anyway. So, but we still have those as requirements for TMM as well. So that was kind of the idea, in that you would get. So a lot of my educational research has to do with, being able to. What's called an apprenticeship kind of perspective of being able to do to learn essentially. So, and I'm a big proponent of being able to do research, right, that would allow you to learn that subject, right? So, so that's the way I really think about how to teach, and so TMM was such an alignment with that, right? So I'm so glad I got into the program, or I was be able to teach in the program. It's funny, it started off as just a year contract actually. So, I got consistently renewed for 3-year contracts up until, and I've recently, both me and Dr. D'Ambrosio are now actually tenure track faculty that are part of the program. So, and we recently got another tenure track in French as well, which is great. So it means that our program's kind of set for the most part. We don't have to worry about losing personnel, for example. So that is something that is kind of ongoing for the most part. I kind of lost track of where I was going with that now. You get at it, right? So what else did you want to know? I forgot now.
So I think that was generally it, like how it started to be a program, and then how like the planning came to be to the shape that it is right now.
So that, and it's taken turns and stuff like that. So there. Yeah, I won't discuss any of the the ins and outs of the the administration and things like that. But right now we're kind of steady at about 80 students coming in per cohort essentially, or per so we usually have about 160 in the program, as you probably already know. And then we grew that from the first time we had about 16. So those
16.
16, they took a chance on us. Then it went to 30 and then 40, and then the pandemic hit and we actually took 80, which is kind of, but we could take them online, so it was easier. So that's what we ended up doing, right? So actually I don't know them as well because I always saw them on screen. So some of them they come to me after, I was in TMM. So I don't recognize them because they were wearing a mask or something like that. So it's kind of funny that way. But so that's kind of the evolution of that. So going forward, it could be that we have more students coming into the program, but I think the first idea that we're going to do is we're going to have a third year of the program as well. So that you start in your second year instead. So that you do a foundational kind of year downtown, and then you come to us for a second, third, and fourth. So that means that you would do like kind of some of the standard courses that you would do downtown normally. You would do them with us, like genetics, biochemistry, and then we move those lab courses that we normally do when you first come into second year as well. So including that the inquiry-based.
We should have that too. It's not fair.
Well, I don't know when the timeline for this is. So, funnily enough, our curriculum is we set it pretty early on, actually, and when we were setting up TMM. So, because that was the idea, but the pandemic kind of made things different a little bit. So, and part of it is that we wanted to, align with downtown a little bit because then because they were thinking of doing a foundational year as well, and then choosing different programs, and we would just been one of those. But I'm not sure if that is still something that would be done. So we might just do it independently. So, but I don't know. I don't know what the plan is anymore.
It's just ideas.
Yeah, it's ideas. They're ready to go though. So we'll see. So we'll. I think there's still a need for that, especially because there's this building itself is going to be adapted to more of a teaching facility when
the other
one is ready.
Yeah. So that's part of it too. So we might end up expanding into here to some extent. So I don't know what the plan. It's hard to predict those types of things and what type of space we'll have, or because they were talking about ripping this building down not that long ago. So because the power issues and things like that. So I don't know if those have been resolved, but that's way above what my base
level of predictability.
Yeah. So when that kind of gets resolved and things like that, and then we might know our path forward as well. So I know students though they probably don't want us to expand it that much. So
why?
I think because you feel like you're, it's part of a kind of a small program.
The numbers really, the amount of people is really good. Like if it goes more than that, then it becomes. I mean, I don't know, because my experience has been either this or 600 students.
Yeah, it's like there's
so
no
maybe,
maybe somewhere in between.
Yeah. So here, the biggest classes that we have is just the 80.
Yeah.
So and then all the other ones get broken down. So maybe even if all the other ones it's about 50 people, maybe it's still fine with 20 in all or more.
So some of those might be modified into French as well. So because we can do that as well. So that's part of the expansion. So I think that was kind of the idea. Originally it was supposed to be 50-50, but I think just the logistics at the University of Ottawa don't really allow that. Because it's about 20% Francophone at the University of Ottawa, and we got about 20% in our cohorts generally, right? So, but we could make individual courses, some of the ones that you were taking 80 in, that would be just offered in French as well. So that is some of the thoughts that might happen. So then the class sizes don't get up too much, I guess, but.
Because the numbers is one of the
I think it's really important to just creating this community of students and professors, particularly starting with the CPHS program and like the little labs of 16 people.
Yes.
Yeah.
So
No, we don't. We have no intention to change that.
Yeah.
So it just might, be moved up a little bit, so that would be your introduction to the whole program. Because CPHS generally is it's more geared to a little bit younger, second year, maybe first year students, to try to engage them in science for retention in the STEM disciplines. So that is still something that we need to do, right? So, and I think it works really well, right? So, and then just that small group of people that you get to know. So, and that's for me too. When I was an instructor, I mean, those are the people I knew all their names, and then it becomes harder, right? So as you expand, right? So, but yeah, so this happens so naturally.
So good, the future is bright, just a lot of possibilities.
Yes.
Yeah, there's a lot of possibilities. I don't know what will happen exactly in the near future, but yeah, so I think TMM, we recently had a cyclical review as well, because most programs have to go through that, and we did very well in that. So extremely well. And CPHS played a major role in that kind of positive review as well. So that it really stuck out as being a unique aspect. And so we, as an undergraduate program, you have to have a little bit of what I call branding, and so that that's part of the brand, right? So that we're one of the only schools in Canada that offers that, and it's required, right? So I think that's that makes us stand out quite a bit, right? So because our competitors are like maybe McMaster Health Sciences, and there's a few others that are kind of a little bit smaller programs, but are kind of geared towards this type of kind of educational kind of strategies and things like that. So.
Yeah, the first TMM semester was definitely a complete different experience coming out of genetics and biochemistry and those kinds of labs as well. We did have the same amount of lab hours and then the second year.
Right.
But then coming to TMM, it's particularly with the CPHs, we're staying even more. At times you're staying at 9 p.m. at night to just finish the thing, and then you can leave. But it's as it's intended. It replicates how a lab sort of works.
Yeah.
And just the pacing of the thing. So it was a unique experience in that way.
And that's good. And I think it's an easy way to ease you in a little bit, because I don't know about you, but you probably went to real labs.
It's not exactly,
and it's quite a bit different, right? So, a little bit different anyway, depending on what lab you went to. But that gives you a different perspective as well, right? So. Originally, actually, we had multiple rotations, for example. That's why we call them rotations. So we did 2, or did we do 3 in the first? No, I think we just did 2, which, gives students a little bit more, diversity in the program, but it becomes harder when it's larger, right? So that's kind of the reason why we just do 1 now. So, but there's advantages to that too, because then you can do a project for a little bit longer. You can see if you like that for your thesis as well, or you want to work with this person for graduate work or whatever it is, right? So, and I think it's an important aspect because you're going to do most of your learning by doing, for the sense is concerned, right? So, yeah.
Well, we are excited for the class that we're going to have in the fall.
If you don't come, that's fine too. No worries.
We're already enrolled.
You're already enrolled.
Thank you very much, Dr. Wheaton. It was a pleasure speaking to you.
It's a pleasure to meet with you both too. Thank you so much.
Thank you very much.
Thank you for listening to this conversation with Dr. Keith Wheaton. If you're interested in his work and teaching, you can find a link to his website in the description.
This is a Dendrite podcast. To support it, please share this episode with your friends and anyone else who might be interested. We hope you enjoyed this episode, and see you next time.